Okay, so the cerebrospinal fluid provides buoyant force. It's a protective cushion, and then supplies nutrients filtered from the blood. Nutrients from the blood diffuse into cerebrospinal fluid, and then that's supplied to various cells and all that. Okay, it's generated in the skull, and like we said, if you produce a liter a day, then you have to absorb a liter a day. Okay. And if you do not absorb as much as is produced, then you'll cause problems. Okay. The cerebrospinal fluid is absorbed in the spinal column. So it's produced in the skull and it's absorbed in the spinal column. So, however, a narrow ventricle, so it's produced there, it has to get to the spinal column to be absorbed. Now, the problem arises because there is a small duct, this aqueduct, that's where it goes from the skull to the spinal column. If that gets blocked and you keep producing fluid, you'll build up pressure. Okay? And that can be a problem. And so that condition is called hydrocephalus. Okay? If fluid builds up in the skull, and this can happen in kids, that problem is called hydrocephalus. Okay, such narrowing can be congenital at birth, arise via tumor compression, or through cyclical gliosis. Okay. So with continued production of CSF, an obstructed aqueduct can lead to pressure buildup in the brain. Okay, so you can have pressure buildup in the brain. All right. So if I don't know if you've had spinal taps, but if anybody has experience with spinal tap, so to measure the pressure in the cerebrospinal fluid, you do one of the things you can do is a spinal tap. You use a saline solution, and you have to do this. So you're measuring the pressure of this column of cerebrospinal fluid. You have to sit erect, okay, and this is a saline solution that you're using, okay. So pressure in the cerebrospinal fluid is measured as shown using a spinal tap with the patient sitting erect, okay. Unfortunately, if there's a blockage in the aqueduct, that the pressure from the fluid in the brain is not transmitted there. And you would like to know if there's a blockage in the aqueduct by measuring the pressure, but the pressure difference is not large enough to be able to tell that. Okay? So this pressure measurement cannot indicate to you whether you have a hydrocephalus or not. Okay? Unfortunately, this method does not detect hydrocephalus because the blockage in the aqueduct prevents the excess pressure in the brain from being transmitted to the spinal column. So these spinal taps are performed for many reasons: to administer anesthetics, to introduce dyes for contrast on an X-ray, and to withdraw CSF for density measurements. Okay, if your spinal fluid is healthy enough to check that, among other things. So once you do a spinal tap, okay, so here is an important thing. You have a total of 125 millimeters of cerebrospinal fluid. Okay, 125 milliliters. Okay, so you have a total of 125 milliliters of cerebrospinal fluid. Okay, so after you do a spinal tap, you might have small leakage of this fluid. So even a small loss is significant. Okay. Loss of the CSF deprives the brain of some of its buoyant force and support of its protection from physical blows, and headaches result. Okay, so you can't lose too much of that. CSF replenishes over a period of 4 to 5 days. Okay. And in the meantime, lying down distributes the remaining CSF evenly. Okay, so if you have a leak there, you want to be lying down. Okay. All right, now let's talk about the eye. All right, so the cornea and the lens, they do not have blood vessels. Okay, so to supply them with nutrients and oxygen, the eye produces an aqueous humor. It produces about 5 ml of aqueous humor is produced daily by the eye. Okay. Aqueous humor is similar in character to the cerebrospinal fluid and carries nutrients to the lens and the cornea of the eye. Okay, so you're producing 5 cc of aqueous humor every day to supply nutrients to the cornea and the lens. Okay. And so we just learned this. So if you're producing 5 cc of aqueous humor every day, what do you have to do? I'm sorry? Yeah, you have to drain 5 cc of that fluid. Okay, after you. Okay, and so if you don't drain that fluid, what will happen? Pressure will build up in the eye. Okay, and here's what happens, and that condition is called glaucoma, and so we'll come to that. Okay, pressure in the vitreous humor. This is the vitreous humor. We'll come to that in a second. Holds the retina flush against the interior parts of the eyeball. So this is pressing the retina against the eyeball. And if that pressure gets to be too large against it, and helps to maintain the shape of the eye, okay. Okay. Excessive interocular pressure is a symptom of glaucoma and can result in the blindness due to deterioration of the retina. If this pressure gets too high on the retina, it'll damage the retina and cause blindness. Okay, so again, let's review this. So you're producing 5 cc of aqueous humor a day. That is to bring nutrients to the cornea and the lens. And if that aqueous humor doesn't drain and get reabsorbed, you'll build pressure, which can lead to blindness. Okay? All right, so now let's look at the details. So that's the big picture. Again, the shape of the eye is maintained by fluid pressure. Okay, so the shape of the eye is maintained by fluid pressure. Okay. And this pressure is between 12 and 24 millimeters of mercury. Okay. It has 2 fluid chambers. Okay. The front chamber contains the aqueous humor, and the rear chamber vitreous humor. Okay. Now this is a jelly-like substance, and this is so it's okay. The rear chamber contains vitreous humor. Okay. 5 cc of aqueous humor is produced daily in the eye. The excess fluid flows out through a canal and is absorbed into the bloodstream. Okay, so 5 cc is absorbed into the bloodstream daily. Okay, and it flows out through a canal. So if the canal gets blocked, then that cannot flow out and get reabsorbed. Okay. So it's similar in character to the cerebrospinal fluid and carries nutrients to the cornea and eye, neither of which have blood vessels. Pressure created by the aqueous humor in the front chamber is transmitted throughout the vitreous humor. So this pushes on the vitreous humor, which exerts pressure on the retina. Okay. Vitreous humor is a jelly-like substance and that does not circulate and is not replenished. So it's not replenished. Okay, so okay. Pressure in the vitreous humor holds the retina flush against the interior parts of the eyeball. Partial blockage of the drainage canal for the aqueous humor results in pressure buildup. So there's drainage canals for this aqueous humor, and this diagram shows you this. Okay, so see this part of the eye is shown so horizontally. Okay, so see the aqueous humor drains through this canal and gets absorbed by the bloodstream. Okay, so if the Schlem's canal gets blocked, then you have a problem. Okay, so if you have a blocked drainage, they can fix that by inserting tubes to let the aqueous humor drain. Okay. Intraocular pressure can rise to as much as 85 millimeters of mercury. Okay, so the normal range is 12 to 24 millimeters of mercury. If your doctor not treated it, the maximum it can rise to is 85 millimeters of mercury. Above this pressure, aqueous humor penetrates arterial walls and is absorbed into the bloodstream, so the pressure rises no longer. Okay. Excessive intraocular pressure is a symptom of glaucoma. Glaucoma may be treated with prescription eye drops, okay, that improve the draining of the fluid, or drugs that suppress the production of aqueous humor. Okay, if you suppress the production of aqueous humor, of course, you're not getting enough nutrients to the cornea and the lens. Okay. Other solutions are insertion of a drain tube. Okay. So anyway, so all your solutions have to somehow provide drainage. Okay. All right, so we learned about the aqueous humor. 5 cc of it is produced every day. Okay, and you got to make sure you have good drainage. Okay, now we'll look at the pressures in the GI system, in your gastrointestinal system. Okay. Food, drink, and waste products moving through 6-meter long digestive tract systems are fluid or are fluids or fluid-like in character. So once you chew your food and stuff like that, it becomes mushy, so it flows like a fluid. And you know that to make a fluid flow, you have to have a higher pressure at one end and lower pressure at another end. Okay. Your GI system is is 6 meters long. So from there, if you unwind all of this, up to there is 6 meters long. 6 meters long is roughly twice the length of this table. Huh? No, about 20 feet. Yeah. 1 meter is 3 feet, so yeah, 3.3 feet. All right, so here's something interesting. This flow is regulated, like I said, any fluid, the flow is regulated by pressure and valves and sphincter muscles in the system. The pressure in the GI system are usually positive and in the range 10 to 20 millimeters of mercury. Okay, so the pressures are in the range of 10 to 20 millimeters of mercury. So stuff flows from higher to lower pressure, of course. The esophagus is an exception. So here's something very interesting. Its pressure is directly related to the thoracic cavity pressure, and it's negative. So the pressure in the esophagus, because it goes through the thoracic cavity, the pressure here is negative. Okay, so the pressure in the stomach is positive. So which way should stuff flow? Stuff should flow from positive to negative pressure. Stuff would flow backwards, and you don't want that. And how do you fix that? You have a sphincter muscle that stops that. Okay, so the esophagus is an exception. Its pressure is directly related to the thoracic cavity pressure and is negative. A sphincter is needed at the junction of the esophagus and stomach to prevent backflow of the stomach fluids. And that's the most common cause of heartburn, okay, when your sphincter muscle is not working properly. Okay. During swallowing, muscle action in the esophagus, so since there is negative pressure, you're trying to push stuff from lower pressure, you're trying to push stuff from lower pressure to higher pressure. So you have to utilize muscles to do that. Okay. So during swallowing, muscle action in the esophagus forces fluid into the stomach, which is at a higher pressure. Okay. So here's a case where you're pushing a fluid from a lower pressure to higher pressure regions with the aid of muscles. Okay. So blockage in the GI system can also cause pressure to increase, sometimes to the point of rupture due to the buildup of fluids. The stomach is elastic, so pressure in it increases gradually. Okay. So if you eat too much, your stomach just expands. Okay. Becoming large only when the stomach is overfilled. Okay. Yeah. So here's an interesting one. The sensation of hunger occurs when stomach pressure is low. Okay. So here's the problem. If this is your stomach and it's full that much, when the pressure is low and then you feel hungry. Okay. Okay. Which can change with eating habit. The pressure is dependent on the capacity of the stomach. Okay. Now, if you consistently overeat and your stomach gets larger and the pressure is low even when the stomach is filled that much, okay, and you start feeling hungry, but you see this much food is plenty for you. So that creates a problem. Okay. The pressure is dependent on the capacity of the stomach, which can change with eating habits. The stomach stretches considerably when a person consistently overeats, and at large stomachs, relative emptiness triggers the sensation of hunger before the person really needs more food. Okay. So it's a problem that can get worse and worse. Okay. Okay. Sometimes some patients have trouble swallowing, and then you can use a feeding tube, okay, and supply nutrients. Okay. So liquids can be fed down through a tube by gravity since pressure in the stomach is not very large. This feeding method is useful for patients who have difficulty swallowing, okay, but are not liable to vomit. So in some conditions, a feeding tube can be, and the feeding tube is going through your nose. Okay, now we'll talk about pressures in your skeletal system. Okay. Now these pressures are quite large. Okay. So you saw that in the eye, the ocular pressures were only about 10 to 20 millimeters of mercury. Okay. Your blood pressure was 120 by 80 millimeters of mercury. Okay. Now the pressure that your bones are bearing are much, much higher. It's about 8,000 millimeters of mercury. Okay, so when you're standing like this, see, so, oops, I don't draw too well, but, okay, so this bone and this bone, that bone is bearing half your weight. Okay, but I'll point out something interesting. So here's, so the pressure pressure is force divided by area, and the force that is being exerted there is half your body weight on each bone. Okay? All right, and that pressure can amount to quite large. So pressure in the skeletal system are far larger than any fluid pressures in the body, being as large as 7,600 millimeters of mercury under ordinary circumstances. That's 10 atmosphere. Skeletal pressure depends on physical activity as well. Yeah. Now here's a question for you guys. If you look at this diagram, so this bone is bearing a pressure of 7,600 millimeters of mercury. So why is this bone shaped like that? So you see at the joint, the area got larger, and that's because you had to have soft tissue between these bones, and this soft tissue cannot bear as large pressure as a bone can bear. So how do you reduce that pressure? By increasing the area. Okay, so the force is the same, and since you had to make this soft tissue last a lifetime, you increase the area so the pressure on it is smaller. Okay, so that's the design. The surface area of the joint is larger than the cross-sectional area of the bone below. This large area reduces pressure at the joint and prevents its deterioration. Okay. So that's a pretty smart design. Okay. All right, now we'll come to part 2. So part 1 is done. I'm going to open the exam for part 1 today. And I'll give you, I guess, 2 weeks to do it since we don't have class next week. Okay, that's a 100-point test on just those slides. Okay, and now we'll do part 2, which will be exam 2 of 7 in chapter 7. Okay. Yeah. Now we'll talk about diffusion. All right, so here diffusion is, so here's 2 types of molecules. There's these red molecules in this fluid. Okay, so this is red dye in water. If you put a drop of red dye, what will happen is all these molecules are moving randomly, and just via random motion, this red dye will eventually spread through the entire fluid. Okay. If a drop of red food coloring is placed into a glass of water, it will slowly spread out into the entire glass. So this is an example of diffusion. Even in the so there's no large-scale flow. Even in the absence of large-scale flow, molecules move about, bouncing off one another and mix continuously. Okay. So temperature is associated with the average kinetic energy of the molecule. Remember, temperature measures average kinetic energy per molecule. So in this room, for instance, oxygen and nitrogen molecules are traveling at about 500 meters per second. 500 meters is from here to Mainland High School. So each molecule here is traveling that fast. They're going from here to Mainland High School in 1 second. Of course, they don't go there because they're colliding with other molecules. Yeah. And they're colliding about 5 billion times a second. Okay. So that's the rate at which these air molecules in this room are colliding. Okay. So these collisions slow down diffusion since molecules cannot travel very far before having a collision. Okay. Okay, why are we talking about diffusion? Do you guys know why we're talking about diffusion? So this is how we exchange material at the cellular level. Okay, so this could be oxygen carrying blood, and this is how oxygen gets to the interstitial fluid. This could be nutrients, That's how nutrients get across. Okay? This could be carbon dioxide. So, and carbon dioxide is going from a higher concentration to lower concentration. So at cellular level, this is how exchange happens. Okay? The thing about diffusion is a slow process, and it doesn't, this material doesn't move large distances. So diffusion, when you want to exchange stuff over small distances, that's how we do that. Okay? So we exchange via diffusion. All right, materials moved by diffusion move much more slowly than the average speed of a molecule. So the diffusion, via diffusion, stuff moves much more slowly. Okay. So the direction and rate of diffusion are described by diffusions by Fick's first law. The direction of diffusion is from a higher concentration region to lower concentration region. So material diffuses from higher to lower concentration. And the rate is directly proportional to the difference in concentration. So if the difference in concentration is large, then you have more stuff moving, okay, than when the concentration is low. Okay. Now, if you look at this diagram, again, now the concentration is even. Is there any more diffusion happening? There's no net flow, but molecules are still moving in either direction. Okay, so there's no net flow of material. Yeah. All right. Okay, so in this diagram, there's a higher concentration here, and there'll be a net flow from here to there. Molecules are moving from here to there as well, but overall, there are more molecules moving from here from the right to left. Okay. So the rate of diffusion depends on other factors besides concentration. Cohesive and adhesive forces also affect the rate of diffusion. Cohesive forces are forces between like molecules, and adhesive forces are forces between unlike molecules. Okay, so for instance, here's an example. If oil is placed in water, okay, you put a drop of red dye, and that diffuses in the glass of water. You put a drop of oil, and oil doesn't diffuse in water. And why is that? Because the oil molecules attract each other much more strongly, and they do not, and the force of attraction between oil molecules and water is not as strong. Okay, so they don't diffuse. Okay, so cohesive and adhesive forces also affect the rate of diffusion. If oil is placed in water, random thermal motion is not sufficient to mix oil and water, since cohesive forces among the oil molecules are larger than adhesive forces between oil and water molecules. Okay? Sugar, however, easily dissolves and diffuses in water, since its cohesive forces are less than the adhesive forces. So sugar will dissolve in water. Okay? Now, so if sugar in warm water dissolves more easily than sugar in cold water, right? And that's because in warm water you're breaking up the sugar molecules. You're breaking up the cohesive force between the sugar molecules, and they're able to more easily dissolve. Okay? Now, surprisingly, in 100 ml of water, 200 grams of sugar can dissolve. And so there's a maximum, and that's the maximum that can dissolve at 20 degrees Celsius at room temperature. Okay. The limit to how much sugar can be dissolved in a specified volume of water depends on the relative strength of the cohesive and adhesive forces. Okay, so those intermolecular forces decide how much it can dissolve. The rate of diffusion increases with temperature. This is because the average molecular speed increases with temperature. Sugar diffuses more rapidly in hot water than in cold, and more sugar can be dissolved in hot water than in cold. All right, so here are a few processes, important processes that you have to be aware of. Okay, So again, whenever you're talking about diffusion, you're talking about stuff being transferred at cellular level, okay? So you're not talking large distances. So this is the last mile problem. So if you want to transport nutrients and oxygen and other stuff over large distances, you have to have a circulatory system. But the last mile to get to the cells, that's when diffusion comes into play. Okay? Transport via diffusion is always slow and is used mostly at the cellular level where distances are small. Okay, the same is true for osmosis and related processes. We'll come to that. Unicellular organisms are use diffusion and related processes exclusively to transport food and waste products in and out of their system. All right, so here are examples. Okay, so here's diffusion, unaided diffusion. A molecule's being transferred from a higher concentration to a lower concentration. Okay, so if this process is too slow, you can have facilitated diffusion. Okay, so you can have molecules aiding you in the diffusion. Okay, so here's facilitated diffusion. Okay, molecules aiding you. Okay, what is happening here? Okay, so in all these processes, diffusion is happening from a higher concentration to lower concentration. Here, see, these molecules are going from lower concentration to higher concentration. Now, when you want to do that, that's active transport. You have to spend energy doing that. Okay? Now, sometimes you can do it with pressure, but oftentimes you have to do it using energy. Okay, so when you want to absorb nutrients, the last bit of nutrients, you have to do this. When you want to transport waste, you have to do this. Okay, so when you want to transport stuff against a concentration gradient, you will use either pressure or you'll have to spend energy. Okay, so when you spend energy, that's active transport. Plants and insects use diffusion to exchange carbon dioxide, water, and oxygen with their surroundings. Okay. Plants consume carbon dioxide and excrete oxygen. Carbon dioxide diffuses into plants, so plants need carbon dioxide. Carbon dioxide will diffuse into plants, into their leaves, whenever the concentration inside is less than outside. Okay, so the concentration of carbon dioxide in the atmosphere is fixed. So when carbon dioxide will move into the leaves only when the concentration in the leaves is smaller. Okay. Similarly, excess oxygen and water diffuses out of plants whenever the concentration is greater than in the surrounding air. Again, concentration in air is constant with just 20% of the atmosphere. So the concentration, oxygen will move out of the leaves to the atmosphere when the concentration of oxygen is higher in the leaves. Okay? Insects consume oxygen and excrete carbon dioxide. So here's how insects both are diffused from. Okay, so insects. Okay, here's how they get oxygen. So they have spiracles. Insects are small, and these spiracles branch out into tracheoles and so on, and so they'll branch out, and oxygen diffuses directly to the muscles like this. Okay, and this is only possible when you're small, so you don't have too many cells to supply oxygen to. Okay. When you start getting bigger, then you have to have other transport mechanisms because you can't have too many of these branching tubes. Okay. Oftentimes you have to diffuse through membranes. So here's a membrane, and so these these molecules, these tiny molecules, there's a higher concentration here, and they're diffusing through this membrane into this site. These membranes generally are semi-permeable. They will selectively let certain molecules and not other molecules. Okay. Most diffusion in biological organisms take place through membranes. All cells and structures within cells, such as the nucleus, are surrounded by membranes. Okay. The membranes are very thin, about 100 atoms thick. So 100 times 10 to the minus 10 meters, that's the size of an atom. Okay, so a membrane is about 100 atoms thick. Most membranes are selectively permeable, meaning they only let certain molecules cross and others not. Okay, so this phenomenon is actually not very well understood. There are several explanations for why membranes are semi-permeable. One is that there are pores in some membranes through which substances can diffuse. These pores are so small that only small molecules get through. Okay, again, like I said, the subject is not completely understood. But in biological systems, you have semi-permeable membranes, so you have to remember that. Okay. Membranes that let certain molecules go through and others, not others. The transport process in biological systems become increasingly more complicated in larger organisms. So even in an earthworm, okay, you can't rely on diffusion alone. An organism as large as an earthworm requires a tube along its length to move food and waste products. Larger organisms have complex systems to transport material in addition to diffusion at the cellular level. So diffusion, you're exchanging material via diffusion only in the last mile. Okay? So you have to have a transport system, again, to transport stuff large distances. Okay, so you eat a meal, nutrients have to be transported from your digestive system through a circulatory system to your cells. Okay. Humans, for example, have many billions of capillaries, veins, arteries, and a host of other tubes in several complicated systems to transport material. Okay. Okay. Now we'll talk about osmosis. Osmosis is the motion of water across a semi-permeable membrane. Okay. So here's a semi-permeable membrane. Let's see what's happening. So here is pure water on this side, and here is a solution. I guess so, something mixed in water. So it could be a sugar solution or whatever, or a salt solution. Okay. So this membrane does not allow sugar molecules to cross across, only allows water molecules to cross across. Okay, so water molecules, this has water on this side as well and water on this side as well. But there are more water molecules on this side per liter than here because there's sugar here. Okay, so on average, water will move from here to there. Okay. Well, how long will it move? Eventually, you'll have equilibrium. So you see, enough water has moved that this level has risen by that much. Now, this excess pressure is preventing more molecules, water molecules, from going across. Okay, and this is called the osmotic pressure. All right. So for instance, if you took that pressure and if you applied that pressure here, you could make force water in the opposite direction. You guys have bought reverse osmosis water from Publix and places like that. Okay, so that's if you have impure water, you can, by applying pressure, you can make water move in here. So you can purify water that way. But you have to do work. Okay, so that's reverse osmosis. Okay, if you apply sufficient pressure. Okay? You guys are aware of desalination plants? Okay, so desalination plant, you're purifying seawater. Okay, and it turns out that the pressure you have to apply is enormous, and desalinating seawater is quite expensive. Okay. Shown here is a solution of glucose water separated by a semi-permeable membrane that allows only water molecules to diffuse through. So here's a semi-permeable membrane that allows only water to diffuse through. Water diffuses from a region of higher concentration to a region of lower concentration. So the concentration of water here is higher than here. Okay. And water moves from a higher concentration to lower concentration. Osmosis is defined as the transport of water through a semi-permeable membrane. So osmosis is transport of water through a semi-permeable membrane. Okay, so in this case, if osmosis is allowed to continue, this situation in B results. This extra pressure on the right, so you see water moved against this extra pressure, and that's called osmotic pressure. Okay. And once H becomes large enough, the back pressure forces water back through the membrane. Okay. So when this pressure is enough, the amount of water flowing this way is equal to the amount flowing this way, and that'll be equilibrium. And that's called osmotic pressure. Okay. Okay, so that is osmotic pressure when equilibrium would reach. Now, if you have solutions on both sides, so let's say here's a concentrated sugar solution and this is not as concentrated, then the pressure is called relative osmotic pressure. Okay, water would still flow from a higher concentration region to lower concentration region. Okay, so if you have, this is a more concentrated sugar solution and this is a lightly concentrated, less concentrated sugar solution, water would move from the less concentrated to the higher concentrated sugar solution. Okay. Osmotic pressures can be rather large. If pure water and seawater are separated by a perfectly efficient semi-permeable membrane that will not pass all, the pressure necessary to prevent osmosis is 26 atmospheres. That is a huge pressure. Okay, so what that statement is saying is, if this were seawater and this were pure water, the column it would eventually rise to is 26 atmosphere, which is 260 meters, which is 3 football field length. Okay, again, the height of that would be 3 football field length. Okay, so what that means is, if you want to desalinate seawater through a membrane, the pressure you would have to apply is 26 atmospheres. And okay, that would cost you quite a bit. Okay, so reverse osmosis takes place when the pressure opposing osmosis exceeds the osmotic pressure. So you can force water from an impure solution through a semi-permeable membrane. You just have to apply greater pressure than osmotic pressure. Okay. Reverse osmosis can be used to desalinate seawater, but is not economical due to the lack of efficient and durable membranes. Okay? So that's one of the problems in desalination. I don't know if you guys know, Tampa has a large desalination plant. It costs a bit of electricity. So osmosis in biology. So here's water in the soil, and this water has higher concentration than the water in the sap, and so water gets into the root via osmosis. Okay, so relative concentration of water is higher than in the sap, and water gets into the roots via osmosis. Osmosis between roots and groundwater is thought to be responsible for transfer of water into many plants. Okay. Groundwater is purer and hence has a higher water concentration than sap in the roots. So water moves from here to into the roots. Okay. There are some plants that grow in salt water. So the question becomes, how does water get from the salt water into the roots where the concentration of water is greater? Okay, so there must be some sort of active transport there. Okay, water and sap is then transferred by osmosis into cells, causing them to swell with increased pressure. Okay. Again, I'm going to remind you, osmosis is transport of water across a semi-permeable membrane. And wherever there's more water, it moves to a region of less concentration of water. Okay. So again, the mystery of how water rises to the top of large trees is still not completely solved. Relative osmotic pressure is not large enough to cause sap to rise to the top of tall trees. To do this, the sap would have to have more dissolved material in it than can be found. Other processes in addition to osmosis are probably involved in the uptake of water. Okay. Some plants grow in salt water that would remove water from the plant if only osmosis were present. Okay, so again, osmosis is transport of water. Okay, so you guys have probably heard of this Epsom salt soak. Okay, so what is the problem that you're trying to solve with the salt soak? And how are you solving it? When do you do a salt soak? What's that? Yes, so let's say if your feet are swollen, there's a lot of water in your feet, and to get this water out, all you have to do is surround it with a solution where there's less water. The water concentration is smaller, and so there's more water here, and it'll come out of osmosis. And to make this less concentration, you just add a lot of salt to it. Okay, so the more salt you have here, the faster this fluid will come up, water will come up. Okay. So if you want to get water out of your legs, inside your legs there's water and salt, but if you surround your legs with a solution, salt solution which has more salt and less water, water from the legs will come out. Okay. So you're using osmosis to fix that problem. Okay, so interstitial fluids. Okay, so these are cells. Okay, and that's the interstitial fluid. Okay, so what happens is nutrients, oxygen, carbon dioxide, and all are exchanged with the capillaries and the interstitial fluids, and then from the interstitial fluids to the cells. Okay. In humans and other animals, the interstitial fluid is regulated by exchange of substances with the bloods and capillaries. Okay, so the interstitial fluid exchanges oxygen, carbon dioxide, nutrients, and waste. So that's what's exchanged between the blood and the interstitial fluids. And how is it done? We diffusion and active transport and all that. Okay, so this is the last mile thing. So all that stuff is exchanged, but we'll just talk about exchange of water. Okay, so this will illustrate some issues. Okay. So what you'll see is when the body wants to exchange some stuff, it has to, okay, so let me show this kidney example. Okay. Okay, so there is waste in the blood. Urea is one example. Again, urea, ammonia, creatine, and etc. So there's waste in this blood that you want to get rid of. Okay. And so what you do is you force out the waste by excess pressure in these capillaries. Okay. When you apply this excess pressure, you're sending out waste, but you're also sending out good stuff. Okay. So this is a nephron in the kidney. You're forcing out the good stuff with the bad stuff. Then what do you have to do? Then you have to reabsorb the good stuff. Okay, and you have to waste energy and spend money to do this. So in this kidney, in this nephron, you're forcing out the good and the bad stuff, and you're reabsorbing the good stuff. Okay. And it turns out that you end up reabsorbing some of the bad stuff as well, but there's a net discharge of the waste. Okay. So you have to be aware of that. Okay. So we'll come to that. So right now, so here's blood flowing, here's a capillary, and we'll talk about exchange of material to the interstitial fluid. Okay. And we'll talk about exchange of water. Okay. Now, okay, so when you're trying to send nutrients and stuff like that, one of the side effects is you also exchange water. Okay. So the overall result, okay. Water is more concentrated in the interstitial fluid than in the blood. There is higher concentration of water in the interstitial fluid than in the blood. But typical osmotic pressure difference between the blood and the interstitial fluid is 22 millimeters of mercury. So the osmotic pressure in this direction is 22 millimeters of mercury. Okay. In the blood capillaries, in the arterial end, okay, so let me show you a diagram. Okay, so the osmotic pressure is 22 millimeters of mercury between the interstitial fluids and the capillaries. And look at this, at the capillaries, the pressure is 35 millimeters of mercury at the capillary end. And at the venule end, it's 15 millimeters of mercury. So I'm going to go back to that diagram in a second. So water from the blood will flow into the interstitial fluid because this pressure is greater than the osmotic pressure is 22 millimeters of mercury. So you lost water when you were trying to force nutrients, oxygen, and all that kind of stuff. What do you have to do now? You have to reabsorb the water. And see, the osmotic pressure was 22 millimeters of mercury. So you have reverse osmosis at this end and you're losing water, but you're absorbing it back at the venule end because of the osmotic pressure. Okay. So while trying to supply nutrients and all, you're forcing out water from the blood, but that is being reabsorbed. Okay. Now here's when you guys know when water collects in your extremities, in your legs and stuff like that, what's happening is, so water will get absorbed if this pressure is lower than 22 millimeters of mercury. That's the osmotic pressure. What if this pressure gets higher than 22 millimeters of mercury? Then the water is not reabsorbed, and that's when water collects in your legs. Okay, so what will cause this pressure to get higher? Okay, if your left ventricle, if you have right heart failure, this ventricle is not able to push blood out, blood collects, and that pressure will rise. So, if fluid is collecting in your legs, that could be a sign of weakening right heart. a weakening right heart failure. Okay. All right. So again, so here's an important thing in your body when you're trying to get rid of transport good, bad, or whatever stuff. There, you're transporting a whole bunch of stuff. Okay. So from the capillaries, let's say you want to supply the interstitial fluid with nutrients and all, you're also forcing out water, and that water has to be reabsorbed. Okay? The osmotic pressure is 22 millimeters of mercury. So when at the arterial end, when the pressure is higher, water is forced out, and this water is reabsorbed at the venule end where the pressure is lower than the osmotic pressure. Okay. All right. So we'll consider the exchange of water. The figure shows the process under average conditions. Blood is a complicated fluid. It contains water, glucose, electrolytes, gases, proteins, red and white blood cells, waste material such as urea, and so on. Blood enters from the top right. So here is the arterial end. And there the pressure is 35 millimeters of mercury, like we saw. And at this end, it's 15 millimeters of mercury under normal conditions. Okay. Blood enters from the top right, and because the capillary is small in diameter, there's a significant pressure drop. Pressure is 35 and 15 millimeters of mercury. Okay. The osmotic pressure is 22 millimeters of mercury. Okay, so water is more concentrated in the interstitial fluid. So interstitial fluid, there's more water than in the blood. And generally, water would want to move from here to there. The osmotic pressure is 22 millimeters of mercury. So if you want to make water move from the blood to the interstitial fluid, you have to have a pressure greater than 22 millimeters of mercury. Okay? So reverse osmosis therefore occurs near the entrance of the capillaries. At the entrance of the capillaries, the pressure is 35 millimeters, and reverse osmosis occurs. Water flows in the opposite direction. Okay. Blood pressure near the capillaries exit is less than the osmotic pressure. Here it's 15 millimeters. of mercury. And the osmotic pressure, which is 22 millimeters, will force the water back. Okay. So water comes out into the interstitial fluid and goes back in there. Okay. Okay, so that's conservation of water, and which is good. Okay. Now, but this excess pressure forces nutrients and other stuff, which is what you wanted, into the interstitial fluid. Okay. The overall result is an exchange of water, while the total amount of water in the interstitial region remains constant. Okay. Okay. When will fluid collect here? Fluid will collect here if the pressure here at the venule end goes from 15 millimeters to 22 millimeters and above. And that happens during right heart failure. Okay. Okay. Departure from this normal behavior can occur for a number of reasons and result in either too much water in interstitial regions or too little. Both conditions are medically unfavorable. Edema or the buildup of interstitial fluid creates swelling in these regions. Okay. The right side of the heart receives blood from the veins. If a patient suffers right heart failure, this pressure can go about 22 millimeters of mercury. Okay. Then the heart is less effective in taking up the blood sent in. Okay. This causes reverse osmosis all along the capillary and the buildup of interstitial fluids. If this pressure rises, water is moving from there to the interstitial fluid. Okay, again, let me go back to that diagram. It's 59, okay. Okay, again, if this pressure is, okay, if you have right heart failure, the ventricle is not able to pump out blood, fluid collects, and the pressure rises all along. And if this pressure gets to be more than 22 millimeters of mercury, water is not being reabsorbed, and water collects in your legs. Okay? And that's edema. Okay, so there's another similar problem with the left heart failure. Okay, so the left side of the heart receives blood from the lungs. Left heart failure increases pressure in the lungs, causing reverse osmosis there. Okay, this water buildup is called pulmonary edema and is an extremely dangerous condition. The patient can literally drown in their own fluids. Okay, so this pulmonary edema happens like so. Okay, so if you have left heart failure, this is not able to this is not able to pump blood out. Blood pressure rises here, and that will in your lungs, and that will force fluid out. Okay, and so you will drown in that. Yeah, so that's pulmonary edema. All right. Okay, so the left side of the heart receives blood from the lungs. Left heart failure increases pressure in the lungs, causing reverse osmosis there. Okay. So this water buildup is called pulmonary edema and is extremely dangerous condition for the patient, and you can drown in that. All right, we'll stop here, and on December 3rd, we will finish this chapter and start the vision. We'll do electromagnetic theory and do vision on December 10th. Okay.